It's a pleasure to be with fellow alumni and to be able to have an opportunity to tell you about some of the things that I'm passionate about. But one of the things that I'm passionate about actually are the students that we have here at Stanford, and Maria is an excellent example of what makes it so exciting to be on the faculty here. She's a bioengineering student, and the bioengineering department here is just doing phenomenal work in all sorts of different areas. But the Medical Scientist Training Program, or the MSTP, which is the MD-PhD program that she's going to be entering next year, is, in my opinion, the premier MD-PhD program in the world, and it's really just terrific to have students like her to interact with as we go through what we're going to be doing. It's a small enough group that if you want to interrupt me if there's something that is not clear, please don't hesitate, and I'd be happy to take questions as we go. I do have one disclosure, and that is that Stanford likes me to point out that I still own some equity in Merck, and so therefore I'm going to be talking about work that Merck is doing, and so that's my disclosure. And here's an outline of what I want to talk about. I'd like to start off with a brief introduction about vaccines, and then talk about how it is that vaccines work at a high level. And then I'd like to introduce you to some of the history of Merck vaccines and a hero of mine named Maurice Hilleman, and then address with you the issue of HIV and what it is that we're trying to do there. You know, vaccines really are just an amazingly cost-effective way to prevent disease. And some of the things that we know about vaccines are, first of all, that this disease caused by smallpox has literally now been eradicated from the earth. And polio, measles, and rubella are actually targeted for elimination, and the WHO is really focusing in on how it is that we can get rid of the last remnants of these infectious diseases. And the agents that are allowing us to do that, and the agents that are allowing us to talk about eradication, are actually vaccines. Indeed, in the United States, there are now 10 infectious diseases that have been over 90% eradicated. So a remarkable statistic, and indeed a very large component of the increase in lifespan in the United States can be attributed to the prevention of death due to infectious agents as a result of vaccines. And of course, vaccines impact not just individuals, which they do, but they also impact whole countries and actually economies. And so what you see when you follow the introduction of vaccines into less developed countries is that as vaccines get introduced and people become healthier, the economies actually improve of those countries. But finally, because vaccines are given to healthy people and often to infants, as opposed to sick people, such as drugs, there can be concerns over possible risks that are associated with them. And as a result of that concern, which of course is very legitimate, vaccines are highly studied in the general population even after they're approved. And indeed, after they're approved, they're studied even more, and they're highly regulated after approval. Unfortunately, there has been some controversy in the field with vaccines, and a large part of it goes to a person named Wakefield. And what Wakefield did was to publish a very influential paper in The Lancet, which is a prestigious medical journal published in the UK in 1998. And what he said was that he had uncovered a correlation between measles, mumps, and rubella vaccine and autism. And unfortunately, the vaccine for measles, mumps, and rubella is given to infants at about the same time that autism first appears in infants anyway. And this then led to really a great deal of concern and a lot of people not vaccinating their children because they were concerned about autism. And as a result of that, there have been outbreaks and continue to be outbreaks of these diseases, and in several cases, deaths, clearly unnecessary deaths due to these diseases because people are afraid to vaccinate their children. But in 2010, The Lancet, this journal, actually retracted the study, and it told The Guardian, which is the newspaper, the famous newspaper in the UK, that it was utterly clear without any ambiguity at all that the statements in the paper were utterly false. And a further investigation by the British Medical Journal actually concluded that this study was an elaborate fraud. Okay? And they uncovered financial ties between Wakefield and the results of this. And they said there was no doubt that Wakefield was responsible. And as a result, the UK's General Medical Council stripped Wakefield of his medical license. They actually stripped his medical license from him, okay, ruling that Wakefield had acted dishonestly and irresponsibly in conducting the experiments that led to publication of the paper. And over the years, many institutions, including the United States Institute of Medicine, the American Academy of Pediatrics, have organized several panels because this is obviously a serious concern and something that we really do want to study extensively. And all of these studies have concluded that there's no association between MMR and autism. So I just point that out because unfortunately, Wakefield's not staying quiet. I mean, he's still trying to promulgate this ridiculous notion. And as a result, we're literally losing lives of infants because people are not getting vaccinated. And it's because of an elaborate fraud. Okay, but now going back and on a more positive note, you know, most of us, or many of us, really don't know about polio, but polio was really a very, very significant medical issue. And as you can see, the number of cases per year here exceeded 20,000. And it was only upon the introduction of the vaccines, the Salk vaccine, which is the inactivated vaccine, and the Sabin vaccine, which was the live oral vaccine, that you saw a dramatic decrease in the amount of polio. And today, of course, we have not seen a case of polio, and it's been literally eradicated. This is an unbelievable chart, and it really, I think, speaks to the power of vaccines if they're done right, and the cost effectiveness. That's right, absolutely. The cost effectiveness of this. And it really, I think, illustrates how you can take a terrible, terrible disease and actually eradicate it. That's a very strong word. You know, there are now just 2 countries left, Pakistan and Afghanistan, where we need to get rid of polio. And the World Health Organization is spearheading an effort, a very concerted effort, and their goal is to have a polio-free world by 2018. Okay, and the way they're doing this is what's called ring vaccination. When you see a case, you come around and you vaccinate all the people in the area around there, and you continue to push that around. And so now Nigeria has just been declared free, and so we have 2 more countries to go. And here are the 10 different infectious diseases that are caused by agents as listed on the bottom that are over 90% eliminated in the United States. And what you can't see, except here in pertussis, is there's a second bar in each of these cases. But it's just so small that you can't see it, okay? And that's because we've done such a good job of eliminating most of the causes, most of these diseases in the United States. And obviously, the United States is a wealthy country, and we have the opportunity to do this. And so what many people in the world are focused on doing is making sure we can do this for the rest of the world. So let me turn to how vaccines work. And I'll do this by first talking about how an infection works. And basically, if we consider the natural response to infection, what happens when we get infected? We get infected with germs, which are usually bacteria or viruses. And when they invade the body, they start to multiply. And that invasion is, as you all know, called an infection. And it's the infection that ultimately leads to illness. But the body's immune system, it fights this infection. It fights this infection 2 different ways. One is to produce what are called antibodies. Antibodies are proteins, and they're made by special cells called B cells. And what these antibodies do is they bind to the germ. So they bind to the virus or they bind to the bacteria, and in turn induce destruction by other cells in the body. So they bind to the germ, and then other cells come along and see that the antibody's there, and they kill the virus, or they swallow up the virus and kill the bacteria. But there's another part of the body's immune system called T cells. And what T cells do is they actually attack cells that are already infected. So these cells run around the body, and if they see a cell that's already been infected by a bacteria or a virus, they go in there and they kill that cell. Okay? And so these are the two arms of our immune system that help us fight infection. But after you clear an infection, so after you no longer have that viral infection or that bacterial infection, your body actually remembers what it had done with the infectious agent. Okay, such that the next time you get exposed to the same agent, okay, memory B cells or memory T cells immediately come to your help and clear the infection. Okay, so the first time through you get sick, your body learns. The second time through, hopefully, your body's already learned, it still has those memory B cells and T cells, and it now goes and attacks the infection before you get sick. Okay, and so here are the B cells, and they produce these antibodies. They secrete these antibodies. They bind to the virus or to the bacteria, and that leads to other cells can kill then these agents. Or T cells, which are so-called cytotoxic. So cyto means cell, toxic means kill. So these cells kill other cells. They kill cells that are infected, and these are the two arms of your immune system. Now, what does a vaccine do? Well, a vaccine is a germ imposter, okay? And so it looks like a germ, it looks like a virus, it looks like a bacteria, but it's actually not a virus or a bacteria. And so your body reacts to vaccines with an immune response, in some cases B cell responses, other cases T cell responses, some cases both. It responds, but this imitation infection, this imposter infection, does not cause illness. But your body produces B cells and T cells as if it were an infection. And then when you get exposed subsequently to the real germ, your body's ready to go and it protects you. And so that's basically what we're trying to do with vaccines, is we're trying to come up with a germ imposter that's a good enough imposter that it'll trick your body into thinking it's infected and will produce the B cells and T cells such that when you get exposed subsequently, you will be cleared. Okay, so now let me just move on to Merck Vaccines. Merck, the company that I used to work for, has a very long history in vaccines, and indeed it's one of the things that I found really quite attractive about working with this company, going back to actually the first commercial diphtheria toxin serum in the 1890s, but then subsequently vaccines, measles, mumps, rubella in the 60s, all the way through. And I had the privilege of overseeing the development of, as was described earlier, Gardasil, which was the human papillomavirus vaccine that prevents cervical cancer, Zostavax, which was a vaccine that prevents shingles in the elderly, and Rototec, which is a vaccine for the prevention of diarrhea in young children caused by the rotavirus. And between Gardasil and Rototec, we're talking in each case about 500,000 deaths per year. In the case of cervical cancer, there are 500,000 women per year that die of cervical cancer. And in the case of Rototec, an equivalent number of babies. And so we're really very fortunate to have these vaccines available, and now the key thing is to make them available, make sure that they get to all the people that need them around the world, which is a tall order. This man, Maurice Hilleman, I had the privilege of getting to know and actually meeting with for lunch every few months. And he was somebody who worked at Merck, and he had an office at Merck and kept on going even though he was officially retired. But as was stated when he died on the front page of the New York Times, much of preventative medicine is based on his work, although he never received the public recognition of Salk, Sabin, or Pasteur. But according to the experts, he saved more lives than any other scientist in the 20th century. Right? Absolutely. He's a hero of mine. And he developed 8 of the 14 vaccines routinely recommended today. Okay? I mean, what a record, right? Measles, mumps, hepatitis A, hepatitis B, chickenpox, meningitis, pneumonia, and Haemophilus influenza bacteria. Unbelievable, right? And these vaccines have virtually vanquished many of these once common childhood diseases in developed countries. And for those of you that are interested, there's a wonderful book about his life published by Paul Offit, who is Chief of Infectious Disease at Children's Hospital, about his life, and it's really a wonderful book. But you know, I think it's just, I'd like to take the opportunity to point out the contributions that this man's life has made to the world and continues to make to the world to this day. Okay, now let's move on to HIV. Here is the problem statement. The United Nations and the World Health Organization's most recent tabulation is at the end of 2014. And they estimate that there are approximately 37 million people living on Earth with HIV. Okay. And if you take 37 million and you divide it by the roughly 7 billion people on Earth, what that means is 1 out of every 200 people on Earth are infected with HIV. Okay. And the remarkable thing is that in the United States and in rich countries, the drug industry has actually done such a great job of coming up with pills and drugs that can combat the disease that today, if you get infected with HIV in the United States, you're more likely to die of cardiovascular disease than you are of AIDS. Okay? It's that good. People take pills once a day and they can live normal lives, and they die from something else. Okay? But unfortunately, for other parts of the world, that's simply not true. And the practical considerations of getting drugs to people in remote areas and the lack of infrastructure and the inability to actually deliver in a cost-effective way these drugs and get people to take them every day has not proven to be a viable solution to the world's problems. And we literally have, for example, in Africa, countries where the governments are run by kids because the adults have all died. And it's something that the United States actually considers, in some cases, national security issues because the governments are just so unstable. And many of us feel that really the only, well, maybe not the only, but the only viable, the near-term solution to this is to come up with an HIV vaccine. And indeed, that's something that people have thought for a long time. And you know, the reverse transcriptase, which is an important component of HIV, the Nobel Prize for that was given in 1975. Immunodeficiency syndrome was first detected in the early 80s. The virus was quickly isolated. And after that was isolated and research started, Margaret Heckler, who was at that time the U.S. Health and Human Services Secretary in 1984, said, "We hope to have a vaccine ready for testing in about 2 years." Okay? Yet another terrible disease is about to yield to patience, persistence, and outright genius. Right? Well, unfortunately, she was wrong. Maybe we don't have enough genius, or maybe the vaccine, the virus is just too smart. But the fact of the matter is, it looked as though this was going to be like one of the other infectious agents, and we were able to just come up with a vaccine and get rid of this. And that was in the 80s, and people have been trying since the 80s, and they've been trying very, very hard. And as you know, we don't have a vaccine. And I'd like to just briefly mention one of the early attempts to come up with vaccines. Here's the HIV virus, indicated with this oval here. And on its surface are 2 proteins called GP41 and GP120. Those are the proteins that are on the virus that allow it to bind to the cell through a cell receptor called CD4. And many of the early attempts were actually going after making antibodies, such as I told you about earlier, against GP120. And the thinking was, well, this is easy. You have a virus that has GP120 on the outside. That allows it to bind to the cell. Just come up with an imposter, a germ imposter, which allows antibodies to be made against GP120, and we'll be done. Well, unfortunately, you can make antibodies against GP120, but the virus rapidly mutates. and it mutates and makes all sorts of changes in its coat protein. In fact, it makes millions and millions of different variants every day in an infected individual. And some of those allow the virus to escape, that is, the antibodies don't bind, and then those continue to multiply and you get continued infection. And so literally many, many years of very, very hard work were spent trying to come up with antibody approaches against the virus, and they failed. And they failed in studies that were conducted, including going through clinical trials in several different avenues. People were so desperate that they tried things that were on the margin, even just to try and get a little bit of efficacy with no results. And indeed, to date, these conventional approaches, which work in other infectious agents such as polio and tetanus, have not worked. There's one trial which has been controversial where they think they have a little bit of protection, but has not been reproduced yet. And so basically, the field has concluded that it's really very, very, very difficult, and so far not been possible, to come up with an antibody approach to stop this virus. And so what the community, Merck and the community, embarked upon many years ago was another approach, and that was to try and come up with ways of tricking the body to thinking that it had an HIV infection so that it would produce T cells, these killer cells. And so I'll remind you, these are the cells that I told you kill infected cells. So instead of trying to prevent the virus from infecting in the first place, what the approach was to try and get the body to produce these cytotoxic cells when you immunize with a vaccine, such that when somebody gets exposed to HIV and their cells get infected, those cells would get killed by the T cells. Okay? And let me just walk you through some characteristics of HIV so that I can walk you through the results with this T cell approach. When somebody gets infected with HIV, what happens is the amount of virus in their blood, which is what's plotted on the y-axis here, increases very quickly. And I'll point out that this is a log plot. Okay, so these are changes in orders of magnitude. So we're going up to 10 to the 8th, 100 million copies of the virus per milliliter of blood. So very rapidly, when you get an HIV infection, the virus replicates like crazy, and you get a very a large amount of virus. And then the immune system kicks in, and it starts to fight that virus infection, and the virus comes down. And then this all occurs in a period of weeks. And then you end up at sort of an equilibrium. That is, the virus is replicating, but the immune system is fighting it and taking it out. And the rate at which the virus is replicating, the rate at which the immune system is taking it out, roughly cancel each other out, so that you end up with what's called a set point. There's a certain amount of virus that's in your blood. And that virus can vary, amount of virus can vary, but it's there. And your immune system fights and does a pretty good job of keeping things in check. But eventually, the virus wins. But this process can take years. But eventually, the virus wins, and then the person develops frank AIDS, and they die. The important thing to note here is that the set point is predictive of disease. That is to say, if you have a higher set point, then the time between when you first get infected and when you eventually succumb and die gets shorter. So if you have a high set point, you're worse off. And so if you can lower that set point, you can actually help things. Yeah, Dave.
So when Americans take medication to deal with it, are they leveling up at the set point?
Yeah, it's great. When Americans take medications, they bring their viral load down to where you cannot detect it with current technology. What's interesting, just to digress for a moment, is if you stop taking those pills, even though you have no virus that you can see, there's a little bit of virus still around, and all of a sudden, it pops right back up. And so you have to keep taking them for your life so far. But no, absolutely right. And so what that does is it lowers their set point so low that basically they don't have to worry if they continue taking the pills about the disease coming on. The other thing about the set point is it influences the transmission, because it influences not just the amount of virus that's in the blood is correlated with the amount of virus that's in, for example, semen. And so the transmission of HIV is decreased if you lower the set point. So if you think about this in terms of the third world, if we could lower the set point, even if we couldn't eradicate AIDS, if we could lower the set point and decrease transmission by a factor of 2, that would be huge, right? And so one of the things that we were thinking is that obviously you'd love to have something which stopped HIV from infecting, but if we could even just lower that set point, that would be a major contribution to what we're trying to do. Okay, so the clinical goals of the program were to lessen the likelihood of persistent infection and importantly, or was okay, right, lead to a clinically significantly lower viral set point, because lower viral set point predicts a delay in progression to AIDS, and lower viral set points are associated with lower rates of HIV transmission. Okay? And so I won't go through many, many years of really hard work by a team that was led by John Shiver and Emilio Amini. Emilio Amini is now in charge of the HIV research at the Gates Foundation, and John Shiver is now in charge of vaccine research at Sanofi Pasteur. But basically what this shows are results from rhesus macaque monkeys, okay? And on the left-hand side are the controls, which were not vaccinated, and the right-hand side were the monkeys that were vaccinated with something called the adenovirus 5 vaccine, which I won't go into, but basically it contains genes from HIV and allows for the production of T cells that will kill infected cells. And what you can see, each of these different colors are the viral loads as a function of time for an individual animal, for an individual monkey. And what you see on the left-hand side in the control group is that these monkeys get infected, they have different peak viremias, peak amounts of virus, and they all then come down and they hit a set point, and different monkeys hit different set points, just like people. But what you see on the right-hand side is that when you have monkeys that are immunized with the vaccine, they still get infected, but now the virus load becomes much lower, and in some animals, the virus load becomes undetectable as judged by the assay that's being used here, okay? Because this being the limit of, this being the limit of detection right here. So this was really promising, and it indicated that in monkeys, using this strategy, we could actually vaccinate animals and not prevent disease. We still had that problem. Okay, not prevent infection, I should say. We still had that problem, but we could lower the set point significantly. And so that led us to undertake clinical studies, and I had the privilege of overseeing this program as we went through humans and then went into, in collaboration with the National Institutes of Health, the NIH, we set up large trials, 2 clinical trials, to study the effectiveness of this vaccine in people. And the study was started in 2004. It enrolled 3,000 people that were at a high risk of acquiring HIV infection. And the primary hypothesis that we were testing was whether or not we could reduce infection and whether we could have an impact on viral load. Okay? And unfortunately, the study was stopped early because an interim analysis showed that the number of infections with the vaccine versus the placebo was not statistically different. In fact, in the vaccine, we had 24 infections out of 741 people. In the placebo, we had 21 infections out of 762 people. And importantly, the set point was not different either. The set point in the vaccine group was 40,000 copies per mil, and the placebo group it was 37,000 copies per mil. And so 3 years later, 2007, the study was stopped, and there were actually 2 studies, and they were both stopped because they both showed similar results. where the vaccine did not prevent infection and did not reduce the amount of virus in the bloodstream of those who did become infected. And so, needless to say, this was a pretty depressing day, not just at Merck, but for the world. And I can say that before I joined Merck, when I was at MIT, I served on a National Institutes of Health committee that was headed up by David Baltimore to actually survey all of the HIV vaccine efforts in the world. And our conclusion was that by far the most promising vaccine effort that was ongoing was the research that was going on at Merck. And if the Merck vaccine didn't work, then we were going to be in trouble. And unfortunately, that's where we're in trouble. And so the other corollary to this is that we've proven, again, very convincingly, that monkeys are not humans. And it's actually a real problem in that we have studies that we conduct in mice, and we've proven lots of times that mice are not humans. But monkeys are closer, and the reason why we went to monkeys is because you wanted to get closer to the human model. But we don't understand enough about the human immune system and the differences between the human and the monkey immune system to know how to get what are called immune correlates of infection. And so here we are. There are two approaches to the vaccine that we've tried. We've tried very hard to come up with antibodies that will bind to the virus and prevent it from infecting. We've tried very hard, and in fact, the National Institutes of Health did a second study after the Merck study with a slightly different vaccine to elicit a T-cell response, and they got essentially the same results as the Merck results. So now in 3 different large studies, we've shown that we're not yet able to know how to do this. Work is still going on, and there are people who are still working hard on trying to come up with a way of coming up with an antibody vaccine. One approach is to isolate antibodies from rare antibodies from some HIV-infected individuals that actually do produce antibodies that neutralize the virus effectively. They're rare, but they exist. And you can isolate them and then characterize those antibodies and ask what is it about those antibodies that makes them special, and then try, and this is the hard part, to engineer something which will elicit that response. That's really, really hard, and so far has not been possible. On the cell-mediated response, people are trying different vectors or different viruses to try to mimic the infection, to try and come up with better ways of coming up with killer responses. But so far, we're really almost exclusively in animal studies. And so I thought I would just finish by talking about some work that we're trying to do, which is actually something that I've been trying to do for a long time now. This started when I was at MIT. It continued while I was at Merck, but it was not the main program because the main program was the one I just told you about. But now that the main program's dead, we're going back to this. And unfortunately, the HIV vaccine field is one that, from a commercial point of view, as you might imagine, does not provide a very good return, projected return to investors. And so therefore, Merck and unfortunately many of the other companies are no longer pursuing HIV vaccine research. And truth be told, I think until the basic science comes up to the point where we have something for them to develop, it probably makes sense for them not to work on it because they're not in the game of just doing basic research. But what I've managed to do is to bring that project into my lab now at Stanford, and that's what I'm working on, among other things, is to try and push this idea. And so I will spend probably just maybe 10 minutes or so to go through that. And the basic idea is that whereas there are antibodies against the GP120 that I told you about that the virus can easily escape from, it turns out that the way in which the virus infects a cell involves a large change in the shape of this protein, whereby the protein that started originally on the virus actually ends up being a protein in both the virus and the cell it's infecting at the same time. And that change in shape is something that we discovered a long time ago and we got very interested in because it allowed for an understanding of how it is that now this protein could cause a change in shape of the membranes of the virus and the cell to allow the virus to infect the cell. Okay? And so just to go back here, whoops, the virus is surrounded by a membrane and the cell is surrounded by a membrane. And in order to infect the cell, these two membranes need to fuse. And it's this process that allows those two membranes to fuse. And an intermediate in that process is this intermediate, which we call the pre-hairpin intermediate, in which the protein is anchored in both the virus and the cell at the same time. Here's the important thing. This part of that intermediate, which we call the NHR, which stands for N-heptad repeat, but that doesn't matter. The NHR is highly, highly conserved. Across all the thousands and thousands of strains that have been looked at, it's about 93% conserved, identical in amino acid sequence. And so therefore, it's going to be harder, which is not to say it won't happen, but it's going to be harder for the virus to escape if we can get antibodies against it. But here's the problem. This intermediate is only present during the infection process. It's not present all the time in the virus like the GP120 is. So on the one hand, we have a region which is much, much more highly conserved than GP120 to go after. On the other hand, we have to get in there and we only have a finite period of time to do it. Okay? But that's what we're trying to do. And it turns out that we and others have made peptides that correspond to the part of the protein that would bind to the N-heptad repeat naturally to form this intermediate. And those peptides inhibit the infection process by binding to the NHR. So peptides, short amino acid sequences of short stretches of proteins that bind to this NHR, this conserved region, can stop infection. Okay? And one of those peptides is actually a drug. One of those peptides is actually a drug, and that drug has been approved by the FDA. It's not commonly used because it's injectable, whereas the pills that people can take are much easier. But for people who are taking the pills and who develop resistance to those pills and have drug-resistant virus, the peptide is actually the drug of last resort, and it's been approved by the FDA. A small biotech company called Trimeris developed it. That was bought by Hoffmann-La Roche, and the drug exists. What we'd like to do, therefore, since we know this works in humans, if you bind the peptide to that region, is we'd like to figure out how to elicit antibodies that would bind to that region and hopefully do the same thing. And as I said, 92%, I lied, sorry. This region is 92% conserved as opposed to GP120, which as you can see is highly variable, 73% conserved. And here are the results with that peptide that's a drug. These are the phase 3 clinical trial results from the New England Journal of Medicine. And the important point for what we're trying to do is it shows that if you can bind to this region, this NHR region, that in humans you know that you can stop infection. And so now the challenge is, how do we get antibodies to do that? Okay. And what I'll say is that over the years, we've developed what we call mimetics. They're basically trying to use protein engineering to design things that look like this part of the protein in this transient intermediate, and then use those mimetics to try and elicit antibodies that will bind to this region and stop infection. And it's proven to be very difficult. And so we went on and actually went on to isolate a monoclonal antibody using a method called phage display, where you express individual antibodies on the surface of a bacteriophage, and then you can reproduce them and isolate the antibody. The important point is this. we were able to identify antibodies which do bind to this region and which do neutralize. And one of them is this monoclonal antibody that we call D5. And what this graph shows is that that antibody, that monoclonal antibody, is capable of inhibiting HIV infection. Although it's pretty weak, this is about 200, 300 nanomolar, which is not very good. But it shows at least that you can get an antibody to stop this infection. And when we solve the crystal structure of this antibody bound to the NHR region, what we see is that indeed the antibody does bind the NHR region. So it is possible to get an antibody to bind the NHR region and stop HIV infection. And if you do so, you can get some relatively broad spectrum activity, which is good, and that indicates that this conserved region is working to our benefit. And so I'll just close by saying that we've gone on to do studies in mice and get antibodies that work, but they're weak. And also now at Merck, we went on to study non-human primates, monkeys. These results here were with a longer mimetic that uses a chemical scaffold to hold them together. And what we see in these monkeys, each of these data points is a different monkey, the results from a different monkey. Before immunization, we don't see any antiviral effect, either using a very sensitive assay, or a sensitive virus. Well, after immunization, what we see is that in 6 out of the 6 monkeys, we're getting weak responses to weak antiviral responses. But I want to emphasize that these are weak. First of all, the titers, as they're called, are very low. They're not things that are ready for prime time. Secondly, we're using a weak virus or a very sensitive assay. But, so I want to emphasize it's very weak, and I don't think that we're anywhere close to, we have a lot of work still to do, but we finally have at least a pinky with a non-human primate where we can measure antiviral activity using this approach. And so just to conclude, the peptide drug that I told you about validates, if you will, the intermediate as an effective therapeutic target in humans. So we know that that peptide works in humans. And the antibody that I showed you demonstrates that we can get antibodies against that intermediate, and such antibodies will be neutralizing. And we finally now have, I want to emphasize, weak neutralizing activity, but it's in non-human primates. So we finally have a foothold. And so now one of the challenges is to identify what it is that we need to do to enhance this antibody response so that we can actually make it something that we think would be good enough for actual testing in humans. And so with that, I'd just like to say that, I graduated here from Stanford in '85, and I was fortunate to get a wonderful position at the Whitehead Institute as a Whitehead Fellow, where I stayed for 16 years on the faculty and had a phenomenal research lab, phenomenal students, many of whom have gone on to just brilliant careers on their own. And so really just an unbelievable group of people. I then had the privilege of spending 12 years heading up research and development at Merck Research Laboratories, where I got to work with just incredibly dedicated scientists, engineers. I have a deep appreciation of the importance of engineering for what it is that we have to do, and physicians who were involved in the work I talked about, vaccine research and development, but more broadly drug research and development. And I don't think it's that appreciated, but you can't do this sort of work without collaborators in academia and in the government and in other companies. It really is. These are huge things. Efforts as we move forward. And of course, and this is particularly true, we have a very dedicated community of patients and subjects, volunteers in the HIV field that participated in the clinical trials. And I thank you for your attention, and I think we still have some time for questions, and so thank you very much. I'd be happy to answer any questions. Right. Thank you, Maria. Back there. There's
one. The common cold.
Yeah. What about the common cold? It turns out the common cold is a very difficult virus to develop a vaccine. People have tried. The rhinovirus, as it's called, actually has ways that it can escape the immune system and has a deep cavity that people have focused on, but so far not been able to come up with a good vaccine. There are, as you probably know, drugs that are some people are trying to develop, but the issue is because most people don't die from the common cold, the drug has to be very, very, very safe before it would actually go into broad use. But it's actually a very difficult problem. It's interesting. So it's a good one, but no answers yet, right?
So one of the things we're hearing a lot about these days is Zika.
Yes.
And I wonder, within the context of what you've presented today, and not to make you the next Margaret Heckler, but what do you think the path of vaccine or other, treatment for Zika is going to look like?
That's a great
My
question.
understanding is with climate change and
Right.
warming, the kinds of mosquitoes that transmit Zika are roaming more widely, so it has the chance of moving into the first world.
Absolutely. So we're very interested in Zika as well as Ebola. We're working on both. And these are really interesting problems. They're scary problems. In the case of Zika, I think it's really caught the attention of everybody because transmission by mosquito is something that gets people's attention. When you have a mosquito that can cause the transmission as opposed to human contact, now you're talking about a very different situation. The protein on the outside of Zika is well characterized now. It's similar to the protein that's on the outside of a different virus called dengue. And dengue virus is also a problem. So far, it's been not possible to come up with an effective dengue vaccine. There is now one vaccine which has been approved only in 4 countries. It's very weakly effective, if at all. And so that makes me think that Zika could be difficult because it's so similar to dengue. And people have been trying hard on dengue for many years. That said, people are just now starting to work on Zika. And so if we're lucky, and I'm wrong, Zika will be something that Margaret Heckler will be able to say will get done quickly, because if it's as simple as making a particle to elicit antibodies against the Zika E protein, as it's called, then I think it's not inconceivable that we could have a vaccine in not 2 years, but in a few years. We'd have to still go through clinical testing and safety, especially if you're talking about administering it to pregnant women. That now introduces a whole nother level of safety issues. But I think, certainly 5 years from now, would it be possible? I think it's possible. But if it's like dengue, then we're not going to have a vaccine anytime soon.
Just a little bit of follow-up on that. I understand dengue sort of breaks off into sort of 4 different
Right.
specific. Is there any evidence that Zika has similar issues where on the surface it looks like one thing, but actually it's many?
Good question. The question is, dengue has 4 different what are called serotypes. And the thing that is really a problem with dengue is if you get infected with one serotype, you're actually more at risk of having serious complications if you get infected by a different serotype than if you had never been infected with the first serotype. And that's because the antibodies that are produced actually recognize a second serotype, but they don't neutralize it, and they actually help the virus infect the cells. And so it's what's called antibody-dependent enhancement. So the antibodies actually work against you. Now the question is there any evidence that Zika also has these different serotypes? And so far, I'm not aware of any evidence for antibody-dependent enhancement with Zika, between Zika viruses, but that's not to say that it couldn't happen. And one of the things that people are also concerned about, because they're so similar in sequence and structure, is whether there's any opportunity for cross-reactivity between dengue antibodies and Zika, which would really be bad because they're endemic in similar areas. So coming back to your original question, though, I think that it is critical that we do research on trying to come up with a Zika vaccine. I also think it's critical that we come up with antibodies that might be useful to treat patients that have Zika infection. But this, mosquito transmission is definitely a real issue. If you look at the case of Ebola, I mean, we still don't have, although we still don't have a licensed Ebola vaccine, although there are some candidates that look very promising. And it's only now that we're starting to get antibodies that can be used to protect monkeys, and it's many, many years after the outbreak. And so, one of the issues that that I'm interested in, and I think many of us at Stanford are interested in, is how can we respond more rapidly to these things, right? Yes, get the microphone there, yeah.
I just have two questions. The first one is, there was a press release by the NIH this week that talked about being able to identify the amino acids of the HIV virus attaching itself to a cell, and that could be used as a potential vehicle for a vaccine. So I wonder if you were aware of that, and if you could comment on it. And then the second question is, what are the different labs, either private companies like Merck or different academic, like your lab here in Stanford, do to collaborate and to share information? Because it seems like the sum of the parts is greater than the total.
Yes, thank you for those questions. I'm not aware what the press release was that came out of NIH. They're doing some terrific stuff though, okay, I'll check that out. They're doing some terrific stuff there, and the NIH, it's the Vaccine Research Center there, is doing some outstanding stuff, and they certainly are doing a lot in terms of the approach that I talked about briefly, where you isolate antibodies from infected individuals that do neutralize these rare antibodies and characterize them. They've been one of the leaders in that area. In terms of your second question about sharing information data, I think this field actually is very good in that regard. And the NIH has set up what's called an AIDS reagent center, which basically you give the reagents to them, and then when they put them online, and when investigators want the reagent, they can just order it through there instead of going to the individual investigator. So it makes it very easy for things to be distributed. If there are IP issues, then the IP agreements are all worked out ahead of time, and so you can get the reagents if you're a for-profit company with the understanding what the IP considerations are. If you're a not-for-profit organization, often the IP issues are less or are non-issues. The databases, there's a database kept at the Los Alamos National Labs of all the sequence information that comes out of these patients and everything. So there's a real accessible amount of information is available. All the structural information is put on databases for everybody to see. So I think it's actually been very good. And unlike some other fields, this is a field I think where people are trying to cooperate. It's just way too hard, and people realize that we have to work together, right? There were some other questions, yes.
Just a very specific question about one of the pathways that you described. So the protein on the outside of the cell, the GP120, I think.
Yes.
You said that mutates very rapidly. At some point, that protein then becomes ineffective, right?
Right.
So how does the boundary work essentially?
Yeah, it's fascinating. It's a fascinating question. What happens is the virus that is predominant when you get infected is the virus that is the most evolutionarily fit. It's the one that's going to grow the fastest. Okay, so in a free environment with no constraints, this virus is the one that's just going to go down the highway. But now you start putting some constraints on it, and you say, well, if we've got an antibody here, and so now that virus is no longer the fastest one, but another virus which has a mutation there now escapes, and it can now outrun the original virus. But if that mutation causes it to lose fitness, then it's not going to be able to run as fast as another mutation, which maybe has less effect on fitness. And so there's a balance between avoiding binding and being able to run fast. And what happens, you can monitor this by sequencing the viruses that are in a person or an animal as you apply this pressure, and you can see the change in the genetic composition of the viruses that multiply. And then if you take off the pressure, so if you remove the antibody, it very rapidly goes back to where it was humming before. And so the simple answer to your question is that it will find a way out, and the most fit virus under those circumstances is the one that will continue to go forward. Now, obviously, you hope that you can squelch that, but so far it's been very difficult to.
What's the order of magnitude on the variance?
The order of magnitude on the variance, it can be many orders of magnitude. So, and so you can have low levels of sort of embers, but that's enough that when the constraints are removed, it'll pop up. The other thing is, the virus mutates so much that what are called second site mutations can pop up in that original one. So you have a mutation which allows it to putter along, but now another mutation occurs that actually compensates and allows this double mutation to go faster, and then you'll see those, right? Completely different protein, right. And indeed, there are documented examples of transmission of such viruses, right? Yes, over here. She'll bring the mic, yeah.
This is less of a scientific question, but just you had alluded to it. Now that HIV in this country has essentially become a managed disease, almost like diabetes, I'm just curious, where does most of the funding for HIV research at the basic science level come from? Does it still come from the government, or is it more private foundations, or just where is just kind of curious what the source of that is?
Great question. The funding for HIV research still is predominantly from the government, from the National Institutes of Health. However, just this year, the National Institutes of Health and Congress have released the mandate that a certain percentage of the NIH budget be dedicated for HIV research. That's no longer a constraint, which is to say HIV research funding is going to go down, okay? And I think it's in my own personal view is that it's in part because in this country, HIV is no longer the problem that it was 20, 30 years ago. And so therefore, Congress sees a need to fund other research in Alzheimer's and things where we still have a real issue. HIV vaccine research has been funded by the NIH, but also by other foundations. Most notably, the Gates Foundation has been a strong supporter of HIV vaccine research. But I must say, I get the impression that folks that have funded HIV vaccine research are getting tired. Okay, there's real fatigue out there, and what I'm seeing is that people are switching vaccine research to other types of HIV preventative research, such as gene therapy or producing antibodies, which is all fine and good, but it's not going to get us a vaccine. And so I think what's happening are the funders are getting, fatigue is the right word, they are fatigued, and so therefore they're no longer funding it as much. And that's a concern of mine, because it'd be one thing if we were close, but we're back to square one. And so that's a concern, right? Yeah.
Actually, my question is related to C. It's also virus disease, say hepatitis C.
Yes.
I think that one's killing power is way how many fold, 10-fold at least, say, to have infected population there.
Yes.
Although we have some medicine these days on the market, my question is, the different medicine, what's their approach to the disease? And also you mentioned about the setting point
Yes.
before. Is this medicine similarly, like currently we can declare, says you cleared from the virus, but
Yes.
the long-term perspective,
Yes, thank
how
you.
much that? You give some insight about that. Thank you.
Thank you. It's a good question. Hepatitis C is a major problem, and there are now fantastic drugs that are either on the market or in development to literally cure people of hepatitis C virus infection. And so today, if one can go on these treatments and literally get cured so that you no longer have the virus in your body. It's a remarkable achievement. And the implication is that some fraction of people that have hepatitis C ultimately go on to requiring a liver transplant. So it's a serious, or cirrhosis. Unfortunately, the drugs are expensive, and hepatitis C is a worldwide issue, just like HIV is. And in many areas, in particular in Asia, hepatitis C is very prevalent. And in many countries where it's not feasible with today's prices to be able to treat those patients. The quest for a hepatitis C vaccine has also been fraught with failures and is actually now not something that's very well being pursued very hard because it's proven to be a very difficult problem. This virus mutates even more than HIV, and it actually causes more variation than HIV, which is the bad news. The good news is that this virus can get cured in some people, can get cleared in some people by the immune system, which is why the drugs work. The drugs help the immune system finish the job of curing it. There's definitely an impact of the genotype of both the virus and the person on the ability to clear it. And we're beginning to understand that genetics that's going on, right? Yeah. Back. Okay.
What is the relationship between management of HIV and its infectivity? And does that generate an epidemiological problem?
When you say management,
Managing HIV through drugs so that it is it's a lifelong disease.
yes.
So if the disease is being managed with drugs, does that do anything to the infectivity?
Oh, I got you. Yes. Yes, thank you. That's a very important question. It dramatically reduces the transmission rate. And so it is, for people that are HIV infected, there are still, the medical recommendations still involve, protective sex, but the rate of transmission is dramatically reduced if the virus is dramatically suppressed. And so if somebody is very well controlled and has levels of virus in their blood that are basically undetectable, the transmission rate is dramatically lowered as well. And that's been an important part of reducing the epidemic in the rich countries. And that's also why people think that we shouldn't give up on trying to get these drugs, especially now that many of them are going generic, to the poorer countries to reduce transmission in addition to reducing progression to AIDS. Right. Thank you. Well, thank you very much. Right.